Geneious Prime has a pile of tools for analyzing DNA data. It does this by letting raw data be turned into visualizations that make sequence analysis easy and intuitive. In this short video, I will try to show you how to do a very simple mutation analysis in less than half an hour. The software welcomes you with a top action bar and a left navigation panel that includes sample alignments, contigs, genomes, and many other DNA datasets for practicing and playing around with. But we are mostly concerned with the local folder here, where we will create our own project folder. Let's call it test, where we can import our sequence data for further analysis. Before we import sequence data, let us first obtain the reference genome to which we will align the sequences in order to detect mutations. You can obtain reference genomes from the NCBI genome browser. Let's get the complete genome of our good old friend Escherichia coli. The database holds one or multiple reference genomes for each of the organisms. It is always recommended to fetch the first one or the one that is latest in terms of update. If the sequence is in the plasmid, you must, of course, download the one for the plasmid. For the human genome, you can simply download the reference genome of the respected chromosome, as downloading the whole genome will take a lot of space and time for analysis. Make sure to download the annotated version of the sequence instead of plain FASTA sequence, as we have to extract the target gene from the genome with the help of the annotations. Since I have already downloaded the sequence, I am moving on to the next step now. Click on the newly created test folder from the left navigation panel, go to Add, Import Files, and choose the AB1 type files from where you have saved those sequences previously. AB1 types of files are the DNA chromatogram files. You can get the job done by importing FASTA. But chromatogram files are recommended as they provide insightful information about the sequences, which we will look into in a minute. Also, import the complete genome that you have just downloaded. The list of sequences appears immediately, where you can check parameters like the length and quality of individual sequences. The percentage Q or the sequence quality indicates how confidently the bases were determined. Hence, the higher the better. A closer inspection reveals that the ends of a sequence often have very dark blue highlighting, which refers to poor quality of the sequence of those particular bases. You can trim those regions before or during alignment, which is especially helpful for longer sequences, or you can simply ignore any anomalies or aberrations in that dark blue region. Although you can align these sequences with the whole reference genome, which is good for sequences of unknown genes, if you already know the sources of the sequences, it is better to extract the gene of interest prior to alignment. Filter and search for the gene from the annotation data of the reference genome and extract that region, giving it a suitable name. Again, look closely at the extracted region. By definition, a functional gene must not have any internal stop codons. As you can see, our extracted region has a start codon at the beginning and only one stop codon, shaded black, at the terminal. Select the chromatogram files along with the extracted reference sequence. Click on the Align or Assemble button from the top action menu and choose Map to Reference. Double check if the reference is correctly chosen. If not, you can always change it later. Set the sensitivity to the highest and iterate up to 25 times for fine-tuning the alignment. Although the more time you iterate, the better it is. For smaller sequences, it actually does not require as many iterations for confident alignment as it requires for sequences of varying lengths and sources. It takes a few seconds to create the alignment, and you can view the aligned position by clicking on the newly created alignment file. Select and zoom into the region of alignment for a closer inspection. The chromatogram view of these aligned sequences again shows the dark blue shading for low quality sequencing in this region, These bases created significantly smaller peaks and thus not of very high quality output. It is wise to ignore these bases. If the base is a mismatch with the reference sequence, it is highlighted. For instance, the base 248 of the sequence changes from cytosine to thymine compared to the reference, indicating a point mutation. Any color other than green at the identity panel at the top indicates a point mutation of varying frequency. Moving left or right, a number of point mutations are observed. Not all of those are present in every sequence though. At the terminal, you can see a number of mismatched bases, but as those are of low quality, we will ignore those. However, not all point mutations cause changes in protein. Some are silent and does not alter the amino acid in the final protein. To detect these, we have to translate the sequences with a suitable reading frame. As we already know that sequence can be read by 6 different reading frames and nature uses only one at a time in most situations. For an annotated gene sequence like we have extracted, reading frame 1 works just fine relative to the consensus or the reference sequence, but in other cases you need to check each of the 6 frames to select the one that has no internal stop codon. All of the frames in this case except for frame 1 creates multiple internal stop codons shaded black, which would cause incorrect translation. Therefore, choose the frame carefully. On top of that, make sure the genetic code is correctly selected. In this case, the bacteria is automatically detected. A frame will add a second line of code below the bases of each sequence. Just like the bases, any aberration from the reference translation will be highlighted. Immediately after translating, you can see the very first amino acid is mutated. But as you are guessing, we will deny this because of quality issue. In the reference sequence, the amino acid at position 83 is serine, while in the sample sequence it is leucine. This mutation at position 284 of the sequences is therefore a missense mutation. There are a couple more missense mutations here and a few more silent ones. Silent mutations are those where the third base of the codon is mutated. Due to the plasticity of the third base of the codon, its mutation often remains silent. We found a bag of missense mutations at the end of our sequence, but as always, we are not counting those. Thank you for your time.